differentiating factors between a prokaryote and a eukaryote, because the nucleoid is what Anjan said is a distinct space in the cell where the genetic information is located, while as we have actual nucleus, which is membrane bound. A lot of this we covered also in the later slides, so that's why I'm not going over things like endospores. There's an entire section on it. This is the size again. He had a question on the sizes if you didn't get that before. So you want to know approximately eukaryotes and bacteria, and then viruses. Okay, so this is a gram-positive cell, a gram-positive membrane, sorry. Okay. For peptidoglycan, there are two things that make up, two elements of it. There's the NAG and the NAM, and they're linked by peptide bonds, and that's anything that has peptidoglycan will have those because that's the components of it. But the teichoic acid is distinctive to gram-positive only. Gram-negative do not have them. And gram-positive have a bigger peptidoglycan portion than gram-negative. So I'll explain things that have to do with that later. But you should know the characteristics very well for gram-positive and gram-negative and the differentiating features. Yep.
You said the teichoic acid is gram-positive?
The what?
Teichoic acid.
Yes, gram-positive only. Yeah. And in another slide, there's like a variation of the teichoic acid. It's like lipopolyteichoic acid, so that's also distinctive. Like teichoic acid and its derivatives are specific to gram-positive. Okay, so this is gram-negative. So gram-negative, you have your cell membrane, and you have your peptidoglycan portion, you have your periplasmic space, and then you have your outer membrane. So we can start from the bottom. The peptidoglycan portion is very, very thin. So that's a differentiating feature. You should have to know that. The periplasm is where enzymes are located that function in like destroying cell debris, things that enter the cell. He mentioned metabolic activities too. Then you have your outer membrane, and on top of your outer membrane you have LPS, which is endotoxin, and that's important in causing septic shock. And you should know that as well. And gram-negative, because they have two membranes, it's very hard for like certain substances like antibiotics or toxins to target them because they have to actually go through two membranes instead of gram-positive, which just has one membrane. So this is just what I was talking about in yeah, over here, this is what I meant. The lipoteichoic acid, that's also specific to gram-positive. But just remember teichoic acid since it ends with that.
Hey. Another word for peptidoglycan is murin, and that was a test question for me before. So just like make sure you know that.
That's in a later slide.
Oh, okay.
How do you spell that?
M-U-R-I-N-E.
You'll see it later. It's in the later slide. And when he says that if you don't fit the above model, it's because you can't stain certain cells that don't have a cell wall, like mycoplasma. So you have to use different tests for that, or because gram-negative and gram-positive is specific to cells that have a cell wall. You have to know the colors, what we talked about earlier, that gram-positive turns purple after gram staining, and gram-negative is red. And this is just a summary of what I talked about. You should know, I'm going to talk about these. You should know why. So lysozyme, it attacks peptidoglycan. So that's why the gram-positive are they have a high sensitivity to it because they have a higher peptidoglycan portion. And if you remember, the gram-negative, their peptidoglycan portion is actually located in between the cell membranes. So it's harder to reach it, too, to target it. The penicillin, it attacks the bonds between N, A, G, I believe. It's in there later. So it also attacks peptidoglycan. So again, the gram-positive are more easily affected by penicillin. And for bacteria, it only inhibits the synthesis of peptidoglycan. So if they're already bacteria present, they're not going to be affected, but it prevents the formation of new bacteria. Complement is the complement is when they're like they form holes in the cell wall. So because gram-negative has such a thin cell wall, they're more easily affected by the complement system since you could poke holes more easily and lyse the cell. And they both have a cell membrane. Gram-negative has two. Okay, I think everyone's familiar with the membrane. Phospholipids, barrier against the world. We had, in the study guide, we had a portion that mentioned the Hopanoids, and those are steroids, and they function similar to steroids in eukaryotic cell membranes, whereas they increase cell fluidity, So they just, like the unsaturated bonds that, make it more, they can move more easily. That's what Hopanoids do for prokaryotes. And everyone should know what the proteins do, the kinds of proteins. You should know this, what cell membranes function for. I think people had mentioned, it's a barrier, transportation of nutrients, exporting waste. There are many different characteristics, just be familiar with it. Okay. Okay, that's what I mentioned before about the peptidoglycan cross-linked peptides. You don't need to know the numbers. He's just stating that gram-positive has much more of a peptidoglycan portion than gram-negative. And the lysozyme, again, it breaks the peptidoglycan. And the pseudomurin, that is structurally similar to peptidoglycan and functionally similar. It has the same function, but the bonds are different. So you cannot, penicillin and lysozymes that target the bond in peptidoglycan, they can't affect the pseudomurin because they have different bonds. Just like what Ajub said about the A, alpha, and beta linkages, different bonds, so different organisms are treated differently.
Can you say that again?
The pseudomurin, that portion?
Did you say that about penicillin?
Yeah, they can't affect it because the bonds are different. It's structurally and functionally similar, and archaea have the pseudomurin. Bacteria don't. And they're not affected by penicillin, which targets the specific bonds. Because pseudomurin is chemically different. This is important. You should know these 4 types of extracellular material that the bacteria secrete. They all have pretty similar functions. For when something's sticky, it prevents desiccation because it's sticky. So the water can't escape very easily. It forms like a membrane. And for capsules, they're very organized compared to slime layers that are very disorganized, but they're almost structurally similar. And capsules, they're very, because they're so smooth, it's kind of like, it aids the cell in moving because it's like lubrication for them. It's just because of the smoothness. And he wants you to know like these kind of components, they help the bacteria against the host immune system, phagocytosis, also because like if you are very slippery, you're escaping phagocytosis more easily. And attachment, adhesion, these function in biofilm formation. I would know if you have a difference, if you list the difference between archaea and bacteria, you should really know the S layers, those are more common in archaea. Okay, so Fimbria and Phyllae in general have some.
Oh, I just want to say, is there a difference between glycocalyx and capsules? Because in the definition of a glycocalyx, it says it is a capsule.
I think the difference, oh yes, I remember. Okay, glycocalyx are sticky. They have a sticky layer. Capsules are smooth, like again, like lubrication. So the sticky layer has different functions, like I mentioned. But they're both all made of the same things, polysaccharides, and S layers, you should remember, are different because they're made out of proteins.
Can eukaryotes have capsules too, or is that
No, it's just prokaryote. Yeah.
Okay.
So Phyllae and Fimbria, they have similar functions. They both function in attachment purposes. Except for the sex pili, which is a form of genetic transfer, like the two cells will be next to each other, they'll make like a tube, and they'll connect, and then you can transfer a plasmid to another cell and get genetic information that way. And you should know that sex pili are different than pili that function in attachment only. You don't need to know type 4 Fimbria, it's just know what Fimbria do, attachment. The flagella, you should know that clockwise is the tumbling, counterclockwise is running. And then chemotaxis is when cells can move towards chemicals or away from chemicals, depending on if it's attractive to the cell or not attractive. So then they'll use their flagella to move and go away. Okay, so this was the endospore slide. Okay, so only formed by some bacteria that you guys know now. They're very resistant, but they're not immune to everything. If you under high enough heat, they will get destroyed, like autoclaves. And then the calcium protects the DNA, and that's why it's important. They have the main part. You want to protect your DNA, so there are a lot of aspects involved in doing that. Sporulation versus germination. Germination is when the cell thinks, okay, conditions are right again, so it's time for me to get, I'll turn on all my machinery. So then it becomes vegetative and undergoes germination. Germination and vegetative are the same thing. It's just germination is a process, while vegetative means alive. And sporulation is when the bacteria become spores. The process by doing so. Just a summary. Okay. So you need to know the differences for this. All of them. Because for this one, he has stress that you should know 1 and 3. So the photolithoautotrophs and the chemolithoautotrophs. You should know the kind of microorganisms that are associated with those. The electron source, energy source, carbon source, that's what 1 and 3 especially. But if you see a name like chemolithoheterotroph, and you should be able to go off of this chart and say, okay, chemotrophs do this, so, and what the litho portion means and what the hetero portion means, heterotroph portion. And that'll help you because if you see, like, if chemo is there, it's organic carbon. If photo is there, then it's CO2. So different words. Does everyone get that? Culture. Define your synthetic.
Could you just give us examples of organisms that are exposed to
Yeah, they're right over here. That's all you have to know.
Okay, so they were in the grass.
Defined is when you know all the ingredients in your media, and then complex is when you don't. You're not sure about the concentrations. Selective is when you're selecting for, yes? Oh, were you just raising? Selective is when you're selecting for a certain type of microorganism or selecting against. For example, in the next slide, you'll see certain culture media that are specific for gram-positive, and they select against gram-negative. And then differential is when you have on a plate, you want to determine between different types of the bacteria that are there. Next slide again, you'll see blood agar differentiates between alpha hemolysis, beta, and gamma. And then, or staphylococcus organisms that are different based on different features that you'll see on the cell. Macroelements and micronutrients, you don't need to know. You should know the bioelements though from the first section. Okay, so for this, you need to know, he has had a question, for example, which of the following is not selective and differential? So blood agar. Or vice versa, you know, which ones are associated with which category. You don't need to know the details of what each one does, but just generally, like blood agar, you have alpha, beta, gamma. So what each of those does. Beta is when it's completely lysed, and then alpha is when there's partial hemolysis, and then gamma is when there's no hemolysis. For the EMB, you should know that it inhibits the growth of gram-positive, and McConkey does that as well. And then the mannitol salt agar, staphylococcus. That's what it's selected for. And then it has also a salt concentration. So it's selective against organisms that cannot stand that kind of salt concentration. And you could determine it's differential because for all of the last three, you're determining between the different organisms that you find on your plate. So the different gram-negatives that you'll see are the different staphylococci that you'll see. So know what a pure culture is. Pretty easy. A streak plate, when you have 4 quadrants, it's not entirely over the plate. A spread plate, you use a glass rod, and you're covering the entire plate. And a pour plate is when you put the bacteria in your agar, and then you pour it into your petri dish, and then so you're not adding it afterwards. It's already there in the warm agar, and then you add it, and it solidifies.